Hello, everyone. We now come to the final section of Chapter 9, Section 9.3, that we will be covering for this unit. And this is on signaling receptors. We are going to take a closer look at how signaling molecules interact with signaling receptors and thereby generate a or trigger an activation of the receptors in order to elicit a response. So your reading assignment for this section is section 9.3 on signaling receptors, which is found in pages 191 to 193 of your textbooks. So signaling receptors are proteins that receive and interpret information that are carried by signaling molecules. So here we will see how signaling molecules bind to and activate cell surface and intracellular receptors to carry messages inside the cell. So before we look closely into the process, please take note of the following learning objectives. The main objective of 9.3 is to be able to relate the binding of signal molecules to cellular receptors in activating cell signaling. And there are six sub-objectives in this section. First is to describe how signal binding can result in activation of the receptor in a cell signaling pathway, and then be able to predict the locations of signal receptors based on polarity or hydrophobicity of the signaling molecules. The third would be to describe the three major types of cell surface receptors. And there's three types: the G protein coupled receptors, the receptor kinases, and ligand-gated ion channels. And so the final three sub-objectives are to be able to explain the mechanism of activation of G protein coupled receptors, to describe the mechanism of activation of signaling systems involving receptor kinases, and then finally to describe the effects of signal binding to ligand-gated ion channels. To get feedback and affirm your understanding of this section after listening to the video, make sure that you answer these self-assessment questions. So first, how are receptors that bind polar and non-polar signaling molecules similar, and how are they different? Second, how can cells respond to external signaling molecules even when the signaling molecules cannot enter the cell? And third, what are the three possible initial steps following the binding of a signaling molecule to a receptor? So first, let's have a look at the underlying principle that connects or relates the signaling molecule and receptors. Regardless of the distance between communicating cells, as we have seen in the previous section, in section 9.2, a responding cell receives a message when the signaling molecule binds to a receptor protein on or inside that responding cell. So for this reason, the signaling molecule is often referred to as a ligand, from the Latin word ligare, which means to bind. Now, the ligand binds to a particular part of the receptor protein called the ligand binding sites. So this is the location on the receptor, the specific location on the receptor to which the ligand binds. Now, there is a recurring principle that we encounter here, which is that the binding of the ligand to the ligand binding site is very similar to the substrate active site binding in enzymes. So they actually operate according to the same principle. And there are three main important features that relate the binding of the signal molecule to the way the receptor behaves. And that first, the bonds formed between the signaling molecule and the receptors are reversible covalent bonds. So, I put that in red. And I would like you to consider the implication of this, the reversibility, and the fact that the interaction is non-covalent. In other words, think, for example, if the situation is that the bonds that are formed are irreversible covalent bonds, what would be the problem that might ensue or that is created by irreversible covalent bonds in terms of the interaction between the signaling molecule and the receptor when it comes to generating signals or when cells communicate to each other. The second critical feature or attribute is that this binding is highly specific. Okay, so the signaling molecule binds only to the receptor with a ligand binding site that recognizes the molecule. And so this is the basis for specificity of communication. Okay, so it is highly specific, and therefore the kind of association between signaling molecule and receptors are very exact in terms of their binding. Although the binding is reversible and non-covalent, yet it is highly specific. So only one kind of signal molecule recognizes a specific receptor. The last attribute is the allosteric nature of the binding. So I hope that by this time you're already familiar with what the term allosteric means. In other words, when a ligand binds to the ligand binding site on its receptor, a conformational change in the receptor takes place, which activates the receptor. This is the key word there, to pass the message to the interior of the cell by triggering a cascade of chemical reactions within the cytosol. So this is tantamount to the case in the enzymes where the substrates binding to the active site can be controlled by a separate site in the enzyme that causes or influences the way the substrates bind. Okay, so this is allosteric control in the enzymes. In a signaling molecule and receptor, the binding of the signaling molecule causes a conformational change in another part of the receptor that activates the receptor to perform some sort of a function, which is usually a chemical reaction. Now, there are two types of signaling receptors, and here in this figure, what we have is what we call as the intracellular receptors. So these are receptors of signaling molecules that are actually found inside the cell. So nonpolar signaling molecules that can pass through the hydrophobic core of the plasma membrane, they don't need a receptor on the exterior side of the cell. So what happens instead is that once the signaling molecules, such as, let's say, a steroid hormone or glucocorticoids, which raises blood glucose, or another example in your textbook is ecdysone, which is a hormone that causes insect molting. All of these signaling molecules are examples of small nonpolar signaling molecules that can easily traverse through or crosses the plasma membrane because of their um, chemical nature and physical nature. So once the signaling molecules, such as these molecules, is inside the cell, they can actually bind to a very specific receptor, okay, or receptor proteins particularly, and they are located in the cytosol, or sometimes they are in fact even located inside the nucleus. Okay, so these molecules can, in fact, these intracellular, I'm sorry, these small non-polar signaling molecules can also traverse all the way through and can sometimes bind to receptors that are found in the nucleus. But in this particular example, the intracellular receptor is found in the cytosol, and upon the binding of the small nonpolar signaling molecules, it causes a allosteric activation of the receptor, and as a result of that, this activated receptor gets translocated or moves into the nucleus and triggers a specific response. So in particular examples that we know of, the active signaling molecule receptor complex, in this particular case, let's say it's a steroid, the steroid receptor complex often acts as a transcriptional regulator. We will study transcription in unit 4, and you will see that this steroid activated receptor bound by steroids can act as transcriptional regulators in order to control gene expression. Okay. So this is an example of communication between small nonpolar signaling molecules and receptors that are found inside the cell. So the receptors are called intracellular receptors. The second type of signaling receptors are what we have already encountered before, the cell surface receptors. So when the signaling molecule is polar, they cannot cross the plasma membrane and must therefore rely on receptors that are found on the cell surface. So here in this figure here, we have a polar signaling molecule, and it has a very specific cell surface receptor here, this fork-like structure here, and typically you can subdivide the part of cell surface receptors into 3 parts or domains. We have what we call as the extracellular domain, where you can find the ligand binding sites, and then there is the transmembrane domain, which anchors the cell surface receptor to the membrane, because cell surface receptors are effectively transmembrane or integral membrane proteins. And there is the cytoplasmic domain, which is this part here, which is of course exposed to the cytosolic part of the cell, or exposed to the region inside the cell. And the binding of the polar signaling molecule to the ligand binding site triggers a conformational change in the entire structure of the receptor. Okay, this is of course, whenever you speak of conformational change, we are speaking of allosteric control or activation. And therefore, as a result of the binding of the polar signaling molecule to the ligand binding site, the receptor gets activated because of this very specific conformational change. And what happens is that because of the conformational change, the cytoplasmic domain becomes altered to perform some sort of function. So this triggers chemical response or chemical reaction. Okay. So this is the fundamental principle by which cell surface receptors get activated by polar signaling molecule. So the common principle here between cell surface receptor and the intracellular receptors are the fact that the molecules that bind to them are very specific, and that upon binding, they activate the receptor through allosteric activation. Now let's pause for a brief moment and assess our understanding of what we just learned by trying to answer this brief question. So here, you strip off all proteins on the cell surface by using a protease. A protease is an enzyme that destroys proteins. Now, when you add a specific signaling molecule, the cell still responds. Now, what is the most reasonable explanation of this? A. The receptor is a ligand-gated ion channel. B. The signaling molecule does not need a receptor. C. The signal can directly activate the second messenger system and does not need to activate the receptor. Or D. The receptor for this signal is inside the cell and the signaling molecule is nonpolar and can diffuse into the cell. So please go to your brief account and try to answer this question. Okay, so by looking at the way signaling receptors behave, we can now try to envision a metaphor or an analogy, a very useful conceptual metaphor for how the receptor behave. As we saw earlier, a receptor is activated after a signaling molecule binds to its ligand binding site. So, in other words, many receptors act as what we call as a binary molecular switches, okay, or a two-state mode switch existing in two alternative states, either an off if it is not active or it's not bound by the signaling molecule, or it is on if it is bound by the signaling molecule. So in this a particular scenario, therefore, what we see as a communication method between cells come about as a result of a response of the receptor as a quote-unquote molecular switch. So we recognize three basic molecular switches or cell surface receptors in eukaryotic cells. And these are first G protein coupled receptors, second receptor kinases, or they are ion channels. So we will have a closer look at each one of these in the next slides. So let's look at G protein coupled receptors. G protein coupled receptors, or sometimes simply called GPCR. You will find this in the literature in the short form. GPCR stands for G protein coupled receptors. So here it is in this figure. And we can identify, of course, the three basic parts of the receptor as we have seen before. There is this extracellular domain, which binds the ligand. And there is several multiple domains, transmembrane domains that span the membrane, and those are called the transmembrane domains. And there is one here that's jutting out. That is, of course, the cytoplasmic domain. Okay, and so what happens is that when the ligand binds the G protein coupled receptor, it actually causes it to interact with a G protein. And the reason why this is called G protein is the G protein actually binds to a small molecule called GDP. Okay. And when it is bound by GDP, this G protein is inactive. And you notice that this GDP is a membrane associated protein, but it is not a transmembrane protein. It's only bound by a small portion of its entire structure on the cytoplasmic side of the membrane. So the binding of the ligand receptor, or the ligand to the receptor, causes this inactive protein to bind to the cytoplasmic domain, which, as a result of the binding of the ligand receptor, has changed its shape. Okay, so by allosteric activation, the cytoplasmic domain of the GPCR, or the G protein coupled receptor, um, uh, caused a conformational change in the cytoplasmic domain, which then is capable of recognizing the inactive G protein. And upon their interaction, then transfers or triggers a second allosteric activation of the G protein. And what happens is that the GDP, okay, um, is comes off, and then this is replaced by GTP. Okay, and as a result of that exchange of GTP for GDP, this G protein is now active. Okay, and this G protein that is active can now transmit the signal to a signal transduction pathway. Which, of course, we will not have the opportunity to go into detail. But if you wish to understand this more closely, feel free to read section on 9.4. But we won't cover that in this unit. Okay, so this is essentially what G protein coupled receptors can do. Okay, so the activated ligand receptor interacts with the inactive G protein that is bound with GDP um, as a result of a conformational change in the cytoplasmic domain, and by their interaction causes a conformational change in the G protein, which then causes the G protein to exchange its GDP for GTP, allowing it to become active in transmitting a signal to a specific signal transduction pathway, which then generates a specific response. And typically, for G protein coupled receptors, they actually trigger fast or short term responses. An example of this would be the adrenaline signaling in heart muscle during fight or flight responses, for example. The second type of signaling receptors belong to what we call as the receptor kinase family of receptors, and this involves the processes of adding a phosphate group or phosphorylation, or the reverse, which is the removal of phosphate groups in molecules, or called dephosphorylation. So you see here in this figure on the right a process where a protein which is inactive in its state gets activated by the process of phosphorylation using an enzyme called a kinase. So the addition of phosphorylation makes this inactive protein active. So this is the again a an illustration of the off and on state of molecular switches through the process of phosphorylation and dephosphorylation. So the addition of the phosphate group in this particular case by the kinase, the enzyme kinase, activates a protein, whereas the reverse process, which is mediated by an enzyme called phosphatase, removes the phosphate group and then reverses the state of the molecule from on to off. Now, receptor kinases are very special type of kinases because, as you see, their organization is that they are integral membrane proteins, and they also have, again, the extracellular domain, the transmembrane domain, and then the cytoplasmic domain. And many receptor kinases are actually dimers. They are made of two polypeptides that actually dimerizes di, dimerizes, sorry, upon the binding of the signal molecule. So this is the signal molecule. Okay, so the binding of the signal molecule to the receptor kinase causes them to dimerize, and the dimerization, this process here is called dimerization. So they associate together reversibly as a result of the binding of the signal molecule. This is equivalent to an allosteric change as well. Okay, it causes the dimerized polypeptides subunit to change conformation and triggering the phosphorylation or of certain parts of the molecules. And the phosphate group that is added to them is a result of the activation of the kinase activity of the receptors as they dimerize together. So when they dimerize together, one of the member of the dimer will phosphorylate the other member. And so this is called what we call as auto or self phosphorylation. Okay, and as a result of this, you now have an active receptor kinase, which can then interact with other components of the signal transduction pathway to elicit a response. And if you want to go through the details of this process, again, you'll have to look at section 9.4 or 9.5, which we will not again cover in this unit. So this is a sufficient understanding for you to appreciate what happens when signal molecule binds to a receptor kinase. I would like to add that the receptor kinase signaling transduction pathway often leads, not always, but often leads to long-term responses such as changes in gene expression. And in fact, we will see an example of this in chapter 11 when we look at the regulation of cell cycle. The third and final form of signal receptors are called ligand-gated ion channels. Now, you may recall in Unit 2 that ion channels are proteins that alter or allow the flow of ions across the plasma membrane. These channels can be opened, in fact, in different ways. So the flow of ions across them can be regulated using ligands. And these ligands actually have specific, again, ligand binding site in this channel. And typically, these channels are inactive, therefore, does not allow any ions to flow. But upon the binding of the ligand to the inactive channel, it causes a conformational change, an allosteric activation of the channel, opening it up, allowing ions to move in. Now, some of these channels are not regulated by physical molecule called the ligand, but some are open up in response to voltage changes across the plasma membrane and are therefore called voltage gated ion channels. And those that are activated by ligand are simply called ligand gated channels. Signaling using ion channel receptors are especially important for nerve and muscle cells because their function depend on rapid change in ion flow across the plasma membrane. And you will have opportunities to study them in the Biology 119. So this ends our coverage of cell signaling in chapter 9. So our next set of lecture videos for Unit 3 is going to be on cell division and cell cycle that is found in chapter 11. So I will see you folks in chapter 11 after this. Thank you.